Hey guys, welcome to the next online lecture that we're doing today. We are going to be talking about cell structure and function. Just a reminder, you guys will have a quiz on this material on January 27th. You are going to be required to know what they look like, so each organelle looks like, as well as what the function of each organelle is. We will review further, and you also obviously doing a cell model of this stuff, so hopefully we cover all the bases and you don't really have too many questions. So let's begin. So we're going to look at the nucleus first. So the nucleus function is to be the control center of the cell. It kind of controls everything that goes on. And the reason it can do that is because it contains the DNA. DNA is the instructions to do anything in the cell. So in order to use the DNA, you need to access the nucleus. So that's kind of how the nucleus, or why the nucleus is the control center of the cell. It's also surrounded by a membrane called the nuclear envelope that kind of keeps all the DNA inside the nucleus. The next organelle that I'd like you to talk about is called the nucleolus. This is the dark spot in the nucleus. The nucleolus its function is pretty simple. It makes ribosomes. So write that down. I don't have a formal slide on it. So the nucleolus makes ribosomes. It's the dark spot inside the nucleus. Ribosomes. Ribosomes' function is basically to make proteins. That's its role. The word synthesis means to build from many parts. So what it does is it basically builds proteins. It's typically found either free floating in the cytosol, the cytoplasm, so the fluid of the cytoplasm, and sometimes it's also attached to internal membranes. For example, which is the next organelle called the rough endoplasmic reticulum. So the rough endoplasmic reticulum is a set of membranes that kind of create a network of channels throughout the cytoplasm. And what's unique about the rough ER compared with the smooth ER is the fact that the rough ER has ribosomes located on it, which kind of gives it its rough appearance because it looks kind of like sandpaper. When the scientists looked at it, they were like, "Oh, that looks like sandpaper. We're going to call that the rough endoplasmic reticulum." And its function is basically to make proteins, to build proteins that are eventually going to be sent off either to the lysosome or out of the cell through the plasma membrane. The next organelle is called the smooth endoplasmic reticulum. This has very few ribosomes attached, so its function is primarily to build lipids or fats that are eventually going to be made into membranes. That's kind of its role. It builds membranes. You could think of it like that. It is also used to detox foreign substances. So if you know an adult that drinks an alcoholic beverage that contains ethanol, which is the alcohol in alcoholic beverages, your smooth endoplasmic reticulum of your cells will go ahead and take that ethanol, that type of alcohol, and it will break it down so it's not poisonous, because normally ethanol is somewhat poisonous, which is why it causes the effects that it does. Here's a picture of it. Again, inside the cell, you almost always see the rough ER outside of the nucleus. In addition, you have smooth endoplasmic reticulum right outside of the rough ER. So for your cell models, you should have the rough endoplasmic reticulum right outside of your nucleus, and the smooth ER should be right outside of the rough ER. Next organelle, it's kind of a fun word, it's called the Golgi apparatus, or the Golgi body. It's a set of different membranes that are used to basically package and distribute materials throughout the cell and even out of the cell. It also can be used to make cell walls for the plants, but mostly it's used to kind of as the UPS of the cell. It's used to package things and then to ship them off to different places, either within the cell or out of the cell. So this is a potential essay question for you guys that you may have. I'd like you to be able to kind of trace and describe the how proteins are produced and where they go if they're about to shipped off. So this is kind of what I would expect for this essay. So here we go. The information that's contained in the DNA is used to make the protein at the ribosomes of the rough endoplasmic reticulum. Once the protein is made, that protein will then be sent off to the Golgi body or the Golgi apparatus. The Golgi apparatus will then package it and into what's called a vesicle, which is like a UPS truck. That vesicle will then move to the cell membrane itself, and the vesicle will fuse to the cell membrane, and it will spit out the protein outside the cell. So again, that would be basically, that description would be what I would expect on like an essay. Again, talking about that the DNA is used to make the protein at the ribosomes of the rough ER. Then that protein will be sent off to the Golgi body, will be packaged, and then shipped in a vesicle to the cell membrane, which then the vesicle will fuse with, and it will then the protein will exit the cell. So that's kind of what I'm looking for there. Next organelle, lysosomes. Lysosomes are kind of the recycling center of the cell. They contain what are called digestive enzymes, that are these chemicals that will break down these complex parts like proteins or fats or whatever other material might be in the cell. It takes those complex molecules and will break them down so they can be reused again. So they'll take a protein, break it down in a bunch of different pieces, which will then be reused to make a new protein. So again, they break down molecules. You can think of it, I think of it best as the recycling center of the cell. We send things there to be broken down and reused again. They can also destroy cells and form matter that the cell has engulfed by what the process called phagocytosis, which we'll talk about next unit or for the next test. But you don't need to know too much about it. This is basically what white blood cells do to bacteria or viruses inside your body. But we'll talk further about that next unit. Again, another picture. I'm going to end it right there. I think that's enough for right now. We'll finish the lecture later. All right, thanks guys. Have a good one. Bye.